Thanks for joining me as we learn about mitosis, how it works, and why it needs to work the right way. This lesson has 4 sections. 1 is the purpose of mitosis, 2 is the phases of the cell cycle, 3 we will discuss mitosis in animals, and 4 we will discuss mitosis in plants. Mitosis has 4 main purposes outlined here. Let's talk about each. Growth. The human being starts as a single cell in the womb. In a matter of 9 months, this single cell will keep dividing to become a full-blown human being in the mother. After birth, the baby will then continue to grow into an adult. None of this would be possible if it were not for mitosis. Wound healing. Every time you fall and scrape your knee, your body needs to heal that wound. Wound healing is an elegant dance between the immune system and the cells that are next to the wound and still alive. These cells will divide and eventually crawl into the empty space and fill it. None of this would be possible without mitosis. Replenishing lost cells. Every day, hundreds of thousands of cells on your skin die and fall off. Here is a diagram of the various layers in your skin. The cells at the bottom are the youngest, while the cells at the top are the oldest. In fact, the white empty-looking cells at the top are dead cells. Every day, cells from the bottom need to travel up. As they travel, they age and they die. Without constant mitosis to replace the young cells at the bottom layer, you would eventually lose all of your skin. Maintaining your immune system. Your immune system consists of many different types of cells, each doing a different function. In order to maintain your immune health throughout your life, your immune system must constantly replace cells that die. Thus, you are able to remember the cold that you caught a few years ago, such that you can recover faster this time than you did before. For this course, you do not need to remember all the different types of immune cells. This is shown just an example of why mitosis is important. Mitosis is one of 4 phases in what is called the cell cycle. The cell cycle is how long it takes for a cell to divide. Mitosis, here represented by the letter M and the red arrow, will be our focus for the rest of this lesson. But it is useful to know a thing or two about what the other phases of the cell cycle do. The G1 phase is when a cell makes a lot of protein in preparation for replicating its DNA, that is doubling the DNA content in preparation for mitosis. During G1, a lot of the organelles also duplicate themselves because eventually they will be divided between 2 new cells. The S phase stands for synthesis phase, here diagrammed in green. The S phase is the phase during which DNA is doubled or replicated, such that when the cell divides, each daughter cell has the same amount of DNA as its original mother. The G2 phase, here represented by the turquoise arrow, is when the cell makes sure that all the DNA has been replicated correctly in the S phase. It also makes more protein that is necessary before the cell enters mitosis. After mitosis happens, each daughter cell has a choice to either re-enter the G1 phase of the cell cycle or to enter what's called the G0 phase. The G0 phase is for cells that want to remain dormant. That is, they no longer want to divide. This often happens when cells commit to doing a certain function for the rest of its life until it dies. For example, a skin cell that has matured to the top layer will die as a skin cell. It has been programmed to no longer be able to divide. Otherwise, it might divide to produce an old skin cell that has a bunch of mutations and just doesn't work right. A key term to know is interphase, as diagrammed in the gray region of this image. Interphase contains G1, S, and G2 phases. It was coined to distinguish the part of the cell cycle that has to occur before mitosis happens. Here is a summary of what happens in each phase of the cell cycle. Mitosis consists of 5 phases, represented by the letters I, P, M, A, T, or IPMAT. This diagram shows the first 2 of those phases, interphase and prophase. Interphase is the phase of the cell cycle that all cells must go through as they prepare to undergo mitosis. In the G2 phase of interphase, the DNA has already been replicated, and so this cell has twice the amount of DNA as it normally would. In this interphase, the nuclear envelope is still present, containing the DNA inside of it, and the 2 centrosomes have not started moving apart and producing microtubules to connect them. Prophase is when the chromosomes condense into these finger-like X-shaped structures. The centrosomes have begun to move apart to different ends of the cell, and they have created microtubules that connect the 2 of them. These microtubules will serve as the railways on which these chromosomes will be pushed and pulled. A key terminology to know is that when DNA is in the form of these condensed chromosomes, the middle region is called the centromere, not to be confused with the centrosome that forms a spindle. The centromere is the middle portion of the chromosome that is strong and tough and serves as a belt that can be pulled on without breaking the chromosome. Some people separate prophase into 2 phases, prophase and prometaphase. Prometaphase is the part of prophase that is right before metaphase. In this phase, the nuclear envelope has significantly broken down, and the chromosomes are now connected to the microtubules, or what's called the spindle fibers, which originate from the centrosomes at different ends of the cell. Once the chromosomes are connected to the spindle poles in prometaphase, the spindle fibers, then push and pull all the chromosomes until they line up in the middle, a region called the metaphase plate. The alignment of chromosomes on a metaphase plate is one of the most amazing things in biology, at least to me, because in order to align all the chromosomes in this middle plate, both poles, both centrosomes, must sense the push and pull pressure to the degree that it knows exactly when all chromosomes are aligned correctly. And it will not allow the cell to enter the next phase, that is anaphase, until this alignment is correct. Once the chromosomes are aligned, the cell then exits metaphase and enters anaphase. Anaphase is the phase in which these chromosome pairs are pulled apart, half going to each end of the dividing cell. Once anaphase stretches out or separates the chromosomes far apart enough, then telophase begins. Telophase is when the one cell then begins to split into 2 cells. And within telophase, a process called cytokinesis happens. Cytokinesis is the pinching of the cell membrane in the middle to separate one cell into 2 cells. Telophase is also the phase in which the nuclear envelope begins to reform and reappear, such that it will eventually surround all the DNA that has been inherited from the dividing mother cell. Cytokinesis is also a very interesting process in that it is the process in which motor machines build a network of scaffold proteins pinching the membrane in the middle such that 2 daughter cells are produced. Cytokinesis in animals is slightly different than cytokinesis in plants. Actually, it's very different, and we will talk about that later on in this lesson. Remember the acronym IPMAT to remember the sequence of phases of mitosis. Mitosis is a very dynamic process. No lesson will be complete without watching an actual video of what a dividing cell looks like. See if you can pick out the different phases that we just discussed in the following video. When teaching about mitosis, we often focus on the movement of the chromosomes, but so much in the cell happens at the same time that it's often overlooked. In the following video, we're going to watch the dynamics of mitochondria in a resting cell and also in a cell that divides. We'll also look at the dynamics of microtubules, the tubes that form the skeleton of the cell. It will be amazing to see how dynamically these molecules and organelles move, as opposed to the static images that we often show when teaching biology. Notice how real mitochondria are actually elongated. Some of them even actually are fused together, in contrast to how they are drawn in textbook diagrams. The dark circle in the middle is the nucleus, in which there are no mitochondria except on top or below. When this video starts, notice how the mitochondria are moving around inside of these two cells. The dark circular space inside each cell is the nucleus, which is not visible in this video. As these two cells divide, note how the mitochondria are evenly distributed in the daughter cells.
In contrast, when all microtubules are labeled with GMP tubulin, the true extent of the microtubule cytoskeleton emerges. Spectacular dynamics of the microtubule cytoskeleton. Note that many, but not all, microtubules in this cell grow from the centrosome. Only the ends of growing microtubules are visible in this experiment. Those that are static or shrinking have lost their GTP tubulin caps and do not bind EB1.
In this video, notice how the green microtubules push and pull the red chromosomes apart during mitosis. This video is an example of when mitosis goes wrong. Notice that the cell has 3 green dots. Those are 3 centrosomes. Normal cells are only supposed to have 2 centrosomes. The result is that this cell creates 3 different poles when it goes through mitosis, resulting in 3 cells instead of 2. Notice that each cell has multiple nuclei. This is a bad sign. Each of these cells will either die or continue on to be very, very misbehaving cells. These multinucleated cells are a sign of cancer. Mitosis in plants is very similar to mitosis in animals, except for a few constraints. Mitosis in plants has the same phases as mitosis in animals: prophase, metaphase, anaphase, and telophase. Plant cells have a cell wall. Because of the cell wall, plant cells cannot pull apart as easily as animal cells. Oftentimes, plant cells, when they divide, they will split into 2 cells, but the overall size of the original cells is kept because the cell wall prevents the original cell from expanding. The presence of the cell wall makes cytokinesis in plants different than in animals. In plants, the plasma membrane or cell membrane does not pinch to separate 2 cells from each other. In plants, a new cell wall must form. This happens when small vesicles containing cell wall material, they gather between the two new nuclei that have just separated in telophase. And what they do is they eventually fuse to form a solid wall between the two new nuclei, resulting in two separate cells. This is a video of actual cytokinesis in plants. Notice the formation of the cell plate that eventually becomes a new cell wall. This is the end of our lesson on mitosis. You now have the foundation to understand why mitosis is important for organisms, how they grow, how they heal, and how they fight off infection. Mitosis is also important for learning how cancer works and how we can control this beast.